Hello, good evening, everyone.
Good evening, sir.
Good evening, sir.
Good evening, sir.
Uh,
Good evening, sir.
Good evening. So, we are in the chapter cardiac muscles. Cardiac muscle में हम लोग heart. In previous class, we discussed about how the various walls or how the layers of heart are there. In the middle layer of heart, it is called as myocardium. Middle layer of heart is called myocardium. Innermost layer is called endocardium, and outermost layer is called as epicardium. And उसके बाहर का जो portion है, that is called pericardium. Basically, two type of pericardium, parietal and visceral pericardium. And then there is a pericardial fluid in between pericardial space. So today we will discuss about myocardium. In cardiac muscle or myocardium, these two can be interchangeable word. जब भी कोई बोलेंगे cardiac muscle या myocardium, ये दोनों word interchangeable है. इन हार्ट में 3 तरह के मसल फाइबर्स होते हैं मायोकार्डियम. जो एक मसल फाइबर वो है जो which forms basically the contractile unit of heart. It forms mainly 99% of it. It forms mainly 99% of the contractile unit of heart. Another type of muscle fibers are they forming the pacemaker tissues. Mainly the SA node and AV node are the pacemaker. These are very small constituents, but they are very significant. In heart, there is SA node. SA node is the main pacemaker. When SA node is not working in some person, then AV node takes over the charge, and then AV node becomes the pacemaker. So SA node and AV node, and also the muscle fibers are forming the conducting system. Also the muscle fibers are the conducting system of heart means all the portions of heart which are transmitting the depolarization wave which are started from the SA node and reaching up to the various fibers. We will read all portions one by one. First, about the first portion, that is the muscle fiber which form the contractile unit of heart. Just making it the full screen. So myocardium are of 3 types, muscle fiber. So muscle fiber which form the contractile unit of heart, or the which form the or mainly which is called as the main cardiac muscles, they are striated and resembles mostly the skeletal muscle. But you know there are so many differences between the skeletal muscle and the cardiac muscle. Cardiac muscle fibers are also bound by sarcolemma, same like the plasma membrane or the cell membrane of the cardiac muscle fiber is called as sarcolemma. It has a centrally placed nucleus, means एक cardiac muscle fiber में एक nucleus होते हैं. But these and their myofibrils are basically embedded in the sarcoplasm, that is in the cytoplasm of the muscle. Sarcomere of the cardiac muscle has all the contractile proteins, namely actin, myosin, troponin, and tropomyosin. There are few other proteins also that are not much significant. That these 4 are very important: actin, myosin, troponin, and tropomyosin. These 4 important muscle proteins होते हैं. Then another thing about this, it is having the sarcotubular system, although same as the skeletal muscle, but there are few differences. That differences we will discuss in the upcoming slides. Sarcotubular system भी होंगे इसके अंदर. Sarcotubular system, just I want to remind you, it is the made up of the L type of cistern or the sarcoplasmic reticulum along with a T tubule in coving of the basically the sarcoplasmic sarcolemma. So there is some tubule like called T tubules and the L tubule. L is also nothing but the terminal cisterns. They having branching, the cardiac muscles, they have the branches. Adjacent cardiac cells are joined end to end by specialized structures. They are known as intercalated disc. इनमें end to end पे cardiac muscle में they are joined by specialized structures. They are called intercalated disc. Then these intercalated discs having the two types of junctions. इनमें intercalated disc में भी दो तरह के जंक्शन होते हैं. एक गैप जंक्शन होता है, दूसरा डेस्मोजोम्स होते हैं. Desmosomes, that is the tight, very small adhering portions which are making a plaque-like structure and which make the tight sealing between the two cell membranes. And another thing is basically the gap junction that allow molecules as well as the depolarization wave to spread from one cell to adjacent cell. So इनके अंदर gap. So because of the gap junctions, one type of cardiac muscle, like the atrial muscle, they behave like a syncytium. That is, they are behave like a multinucleated structure. They contract and relax simultaneously. So that is called functional syncytium. So heart functions as syncytium. इसमें भी दो important है. When one cardiac cell undergoes an action potential, the electrical impulse is spread to all other cells that are joined by gap junctions. So because of gap junctions, heart function as syncytium, and that syncytium means the multinucleated cells behave like a single cell. And so when they become excited and contract as a single functional unit, एक functional unit की तरह ही वो contract या relax होते हैं. There is atrial syncytium and ventricular syncytium. Means atrium एक साथ contract and relax होंगे and ventricle एक साथ contract and relax होंगे. So this is the basically few special features of the cardiac muscle, and all the features which are noted here in this slide is different than the skeletal muscle. Skeletal muscle और cardiac muscle में skeletal muscle में ये कोई भी features नहीं होंगे. Means उनके अंदर intercalated disc नहीं होंगे, gap junctions नहीं होंगे, they do not behave like the syncytium. So ये typical कुछ differences हैं. This is the structure you must have gone through, that the intercalated disc, branching or the cross striations, nuclei in the and the myofibrils, and there is desmosomes which are connecting the two adjacent cells, and there is some gap junctions between the two connecting cells, so that the cardiac muscle fibers स्ट्रक्चर वाइज इस तरह से दिखेंगे इसके अंदर. रिगार्डिंग मोर डिटेलिंग ऑफ द स्ट्रक्चर, थोड़ा सा और डिटेल में जाएंगे तो देयर इज वन अनदर थिंग दैट आई वांट टू डिस्कस is not about the details of the structure but about the sarcotubular system. Means sarcotubular system of the cardiac muscle को मैं थोड़ा ज़्यादा ध्यान से आपको बताने के लिए ट्राई करता हूँ. Sarcotubular system in skeletal muscle and in the cardiac muscle are near about same. There are some differences in that. What are the basic differences that I want? First, you see the sarcotubular system is what? It is a membranous structure around the muscle fiber. Muscle fiber के around membranous structures होते हैं, which are made up of the terminal cistern, and there are tubules, transverse tubule and terminal cistern के, and there is sarcolemma के अंदर कुछ terminal cistern and tubules which they make. T system or the transverse tubules are nothing, are the continuous with the sarcolemma. It function in rapid transmission of action potential from the cell membrane to all fibrils. Sarcoplasmic reticulum has two enlarged cisterns, two enlarged endings in close contact with the T tubules at the junction between A and I band. There is one another thing that in the cardiac muscle, there is only one sarcoplasmic, that is one cistern are making connection with the T system, one L system or the one terminal system, whereas in the skeletal muscle, there are two terminal systems which make the contact with the T tubule. So जो इस slide में यहाँ लिखा है sarcoplasmic has the two enlarged, it is about the skeletal muscle. But in the cardiac muscle, they are having only one is to one ratio. That is one cistern and one T tubule. So this is one difference between the this terminal cistern, or you can say that the sarcoplasmic cisterns, they contain calcium ions, and the function of sarcoplasmic reticulum is to move the calcium and muscle metabolism. So this is about the few important features of the sarcotubular system. When we look at the sarcotubular system of the skeletal and cardiac muscle, there are few differences. One is the main difference is the diameter of the T tubule. The T tubule are more in diameter, about 5 times more as compared to skeletal muscles. Means थोड़े चौड़े होंगे T tubules, and because of that there is a dyad feature, that is association between the T tubule and cistern, that is one tubule one cistern, whereas in the skeletal muscle it is very thin T tubule, so that the two terminal cisterns are very close to each other, so there is one tubule and two cisterns are together, that's why they form the triad. So sarcotubular system in skeletal muscle is like a triad, whereas in the cardiac muscle it is a dyad, because of basically the thick tubule and narrow tubular cistern. And whereas in a skeletal muscle there is a dilated cistern, dilated sarcoplasmic reticulum, and the uh, thinner T tubules. Sarcomeric organization is less regular in the cardiac muscle as compared to the skeletal muscle. This is basically the diagram showing you the sarcotubular system in cardiac muscle. What I told you in the previous slide, that there is a dyad feature, means the T tubule and the uh, sarcoplasmic reticulum or the cistern portions, there is 1:1 ratio. So this is basically the main feature of the sarcoplasmic reticulum or the sarcotubular system of the cardiac muscle. Now just look at the what are the written in this slide. You can notice that number one point, it shows that when the action potential of wave reaches up to the T tubules, up to the deeper portion of the T tubules, in the T tubule there is a calcium channel. These channels are nothing but this the are also called as the DHP receptors. This DHP full form is dihydropyridine receptors. This is very important receptors, and uh, this on the action on this receptor, there are few drugs are called calcium channel blockers, which are basically they are the dihydropyridine type of blockers. This you will read in the next year pharmacology classes, that how the calcium channel blockers they are used for the treatment of the cardiac disorders, mainly like blood pressure management. So that you will learn arrhythmia treatment, that you will learn in the pharmacology next year. Here just I want to tell that the DHP receptors are present on the membrane of the sarcolemma, basically on the T tubules. This DHP receptor are making a mechanical coupling. You can see here they are making like a mechanical coupling with another receptor which are present on the sarcoplasmic uh, or you can say the cistern portion of the sarcoplasmic reticulum, and that receptor is called as rhinodine receptor, RYR. Here there are two receptors, one is DHP receptor and another is rhinodine receptor. When there is a mechanical coupling, it makes like a tubule like a structure, again another tubule. So T tubule and another it behaves like a tubule here, that is DHP and the rhinodine. So when the action potential reaches here, if there is a voltage sensitive calcium channel, this causes opening of the calcium channel here, opening of the calcium channel present in the DHP receptor, and this causes the entry of the few extracellular calcium. This entry of the free extracellular calcium is the main stimulant here for the muscle contraction of the cardiac muscle contraction. It is not the in the skeletal muscle, that is altogether different. It is not the entry of the calcium channel. Here the single calcium entry, this when enters into the sarcoplasmic reticulum, this causes more release of the calcium, and this is called calcium spark. So there is a more release of this due to this calcium. There is more release of the calcium ion in the sarcomere, in the basically in the sarcoplasma, and this calcium binds with the troponin C subunit, and which causes the uh, opening of the lock between the actin and tropomyosin, and so that there is a muscle contraction is possible, so that the attachment of the myosin with the actin is possible. So this binding of the calcium, this calcium is returned back to the terminal system by another pump that is called Sarca pump, SCRCA. Sarca SCR is the sarcoplasmic endoplasmic reticulum calcium ATPase pump. So this is basically the whole sequence of event. You can see first one, the action potential wave reaches in the cardiac muscle, in the membrane of the cardiac muscle. Then there is the entry of the calcium because of the activation of the DHP receptor, which act like the calcium channel. Then there is a mechanical coupling between the DHP and the rhinodine receptor, and so that this calcium can enter into the sarcoplasmic reticulum, and this leads the more release of the calcium. So this release of the calcium induced by calcium is called as CICR, calcium induced calcium release. Please note this. This is very important. So release of the calcium is because of the some small calcium that causes the more calcium to be released. This more amount of the calcium which are released, they go and bind with the troponin, and thus they allow the actin and myosin to interact with each other, and muscle contraction takes place. The calcium which are present here, which return back to the sarcoplasmic reticulum via another pump that is called endoplasmic reticulum calcium ATPase pump. Obviously, it is ATPase pump, so it needs energy. So it is the energy dependent process here. So the relaxation process where the calcium is returned back is basically energy dependent process. So this is about the whole sequence of event of the excitation contraction coupling in the cardiac muscle. Although I will repeat this slide again, but for you, that is how the excitation causes the contraction in the cardiac muscle. There is the role of the DHP receptor, rhinodine receptor, and once the contraction is over, the calcium which is returned back to this is called the via the Sarca pump. So these three are very important things you should notice and you should remember. And once the calcium is available in the sarcom near to the muscle fibrils, that calcium binds with the troponin C, and that causes the actin and myosin to interact. So this is about the whole sequence of event at the sarcotubular system in the cardiac muscle. This is basically the slide showing you the same thing, basically how the DHP receptor and how the rhinodine receptor channel, they causes the calcium spark, and ultimately the calcium signal forming the muscle to contract, and after contraction during relaxation, that calcium is returned via the calcium ATPase pump to the calcium stores, and this leads to some excess of the calcium which are coming out of the cell membrane in response of the sodium. So sodium entry calcium, there is a sodium calcium exchanger, and which this sodium is returned back again to the extracellular system in response of the sodium potassium ATPase pump. So there are two another pumps that is present interacting with the extracellular, that is the calcium sodium exchanger and the sodium potassium ATPase pump. So there are so many things are simultaneously shown in this picture. You can notice here, and how this is basically the mechanism of the cardiac muscle excitation and contraction. Whole sequence of event are written in the form of, you can see the flow chart where it is written when the action potential enters from the adjacent cell. Point number 1. You can see that number 2, that is voltage gated calcium channel opens. What is the name of this? This is basically DHP receptor. This causes the calcium ions to enter the cell. Once the calcium ion enter the cell, There is because of the mechanical coupling. There is because of mechanical coupling, basically the calcium ion enters the sarcoplasmic reticulum. This causes more calcium induced calcium release that I told you in the previous slide. It is called CICR, calcium induced calcium release through rhinodine receptor channels. Now the calcium which are released, they causes the local spark. Local release causes the calcium spark. When there is a sufficient amount of the calcium spark, it basically summed up, and that it creates the basically the calcium signal. And this calcium signal basically it binds to the troponin to initiate the contraction. The rest portion of the how the calcium binding with the troponin C, it causes the actin and myosin to interact. This will be taught later on. And just at this point, I am leaving this. Now, when the contraction process is over, when the relaxation occurs, then the calcium unbinds from the troponin C. Another calcium which is released from the troponin, they are pumped back into the sarcoplasmic reticulum for storage purpose. And here there is another pump that is called Sarco pump, sarcoplasmic endoplasmic reticulum, ATPase pump. Now the calcium is pumped back into sarcoplasmic reticulum. Some calcium is exchanged with the sodium, and the sodium gradient is maintained by the sodium potassium pump. So this is the whole sequence of event of the mechanism of cardiac muscle contraction and relaxation. Particularly, it is not the contraction relaxation. It is basically how the role, what are the role of the sarcotubular system, and what are the role of the various ion channels which are involved in the contraction and relaxation of the cardiac muscle. So the question can be generated from this slide. Explain the mechanism of cardiac muscle excitation and contraction along with the relaxation. So this can be asked as a question, that is called the excitation contraction coupling in the cardiac muscle. In a skeletal muscle, just you can see there is one difference that I told you, that in a skeletal muscle there is a triad in reference as compared to the diad. Because of the T-tubule is very thin, so there are the two terminal systems are very close to each other, and the ion channels are again the same, the dihydropyridine receptors, L-type calcium channel, and rhinodine receptors. But here it is not the mechanical coupling, here it is the electrical coupling. What is electrical coupling basically? Or you can see the electromechanical coupling. It is basically when the action potential reaches this the Dhp receptor, this causes the opening of this calcium channel. Here there is no entry of the calcium, you can notice here, like in the cardiac muscle, but in a skeletal muscle, it opens this channel. Opening of the channel, it is not again the rhinodine receptor. This rhinodine receptor channel will open, but it is in response of the voltage sensitive. So it is called electrical coupling. It is not the mechanical coupling like in previous case, not making like a tubule system, like they are not. सो इसके वजह से ये गेट खुल जाएगा और यहाँ कैल्शियम रिलीज होंगे. ये सिर्फ कैल्शियम रिलीज का जो प्रोसेस है, इट इज नॉट द कैल्शियम इंड्यूस्ड कैल्शियम रिलीज. ये ऐसा नहीं है, इसमें बेसिकली वोल्टेज गेटेड calcium release. So this is the only difference in the excitation contraction coupling. And after that, there is most of the things are same. That is the same. The calcium will bind to the troponin, then causes the muscle contraction, and that after unbinding, they return back to the calcium storage. Also, there is no sodium calcium exchanger in the plasma membrane जो कि इसको मेंटेन करते हैं, क्योंकि वहां पे कैल्शियम, थोड़ा सा भी कैल्शियम बढ़ेगा या कम होगा तो कार्डियक मसल पे असर पड़ते हैं, बट यहाँ पे उतना ज्यादा असर नहीं है, यहाँ पे ज्यादा सोडियम चैनल का रोल. So these are the few important differences in the skeletal and cardiac muscle. Regarding cardiac muscle fibers, the orientation of muscle fibers are basically they are not in the same direction. Cardiac muscle heart has to contract in different direction. Atrium is contracting this way, ventricle contracting this way, and they are not all the same time. So they are not in the same direction. They are having the different directions. Although they are striated, they will only contract along their long axis. However, in order to get the contraction in two axes, the fibers are here fibers. They are fibers. The fibers are wrapped around. These fibers will wrap around each other. कभी कभी एक दूसरे के ऊपर लेयरिंग बना के वो एक इस तरह से बना लेते हैं जिससे कि वो अपने लॉन्ग एक्सिस में कॉन्ट्रैक्ट करेंगे. So this is something different as compared to the skeletal muscle. So the orientation of the muscle fibers are, although they work in the same one direction, but they are basically the wrapping of the in the two axes. Then another type of the cardiac muscle, another type of the muscle fiber or in the heart, they are forming like the pacemaker. So the एक तो ये हो गया मेन मसल फाइबर, जो कि जब भी हम कार्डियक मसल के बारे में बात करेंगे तो 99% of the cardiac muscle are the main contractile unit. Other than that, there are some modified cardiac muscle. They modified to form like the pacemaker. What do you mean by pacemaker? It means they behave having its own depolarization एंड रिपोलराइजेशन, अपने आप ही बिना किसी स्टिमुलस के, बिना किसी आउटर स्टिमुलस के यदि कोई मसल अपने आप ही, सो दैट प्रोसेस इज कॉल्ड ऑटोमेटिसिटी. ऑटोमेटिसिटी. और यदि वो रिदमिक मैनर में हो तो दैट इज कॉल्ड ऑटो रिदमिसिटी. तो हार्ट के अंदर ऑटोमेटिसिटी या ऑटो रिदमिसिटी बनाने का जो प्रॉपर्टी है, इट इज ओनली ड्यू टू द पेसमेकर. The sum of the cardiac fibers are modified into a specialized structure known as pacemaker. These pacemaker fibers have very less striations. They are called pacemaker because their cells are called as P cells. इनको pacemaker cell या इन्हीं cell को P cell भी बोला जाता है. SA node and AV node, they forms the pacemaker in the human heart. When you read about the conducting system of heart, in that we will see about the pacemaker tissues more detail. Another type of the cardiac muscle, they are forming the conducting system of heart. तो इसके अलावे conducting system of heart, जिसमें कि अभी मैं बोल रहा था कि पेसमेकर टिश्यू का जो भी इम्पोर्टेंट है तो वो भी इसके अंदर आएगा. कंडक्टिंग सिस्टम ऑफ हार्ट इज मॉडिफाइड बाय द मॉडिफाइड कार्डियक मसल फाइबर्स. The impulses from the SO node are transmitted to the atria, and once they reach the atria, they concern back to the AV node, and from AV node to the bundle of his, and from the bundle of his to the right and left bundle branch, and left and right bundle branch, and उसके बाद जो है so they will go to the various ventricular fibers. This is basically the conducting system of heart. This we will again repeat in the upcoming class, that is the conducting system of heart. It starts from the SA node to atrial syncytium. It means to the various atrium, various fibers of the atrium. From there, the junctional fibers, and that is from there to the AV node. So SA node to AV node, in between there are interatrial pathways. Nowadays, earlier जैसे मैंने कल प्रीवियस क्लास में बताया था कि there are three main names. यहाँ पे आप कहीं पे आपको ये इंटरनोडल पाथवेज के 3 अलग अलग नाम भी मिलेंगे. That is anterior, Batchman, Thorel and Wenckebach. लेकिन these names nowadays it is thought that it is not the basically the individual fibers are not found in the heart. डिसेक्शन जब करोगे तो इसमें कभी भी आपको इंडिविजुअल फाइबर्स नहीं मिलेंगे. सो द एट्रियम में एट्रिया विल हैव ओनली बेसिकली द ऑल एट्रियम बिहेव लाइक द सिंसाइटियम. फ्रॉम देयर फ्रॉम द एवी नोड देयर इज एवी बंडल, then they are divided into 2 branches, right and left branches. Right branches through the Purkinje fibers, and the left branches again to the Purkinje fibers, and finally the ventricular syncytium to the all the ventricles. So this is basically the conducting system overview. So the cardiac muscle या हार्ट के टिश्यूज बेसिकली इस तरह से डिवाइड हैं कि 3 तरह के कार्डियक मसल हैं. एक वो जो कि खुद कॉन्ट्रैक्ट करते हैं, दूसरे वो जो कि पेसमेकर टिश्यूज है, और तीसरे वो जो कि कंडक्टिंग सिस्टम के पार्ट है. तो जब हम कार्डियक मसल की बात कर रहे हैं तो हम 99% उस बारे में बात कर रहे होते हैं, लेकिन ये these are also the conducting system as well as the pacemaker tissues. They are also the cardiac muscles. They also are the myocardium. About the properties of cardiac muscles, it is having the 2 important properties. One is electrical and mechanical. Electrical property is excitability, autorhythmicity, and conductivity. Mechanical property is contractility, refractive period, staircase, and Treppe phenomenon. These properties of cardiac muscle we will read in next class. We will have our next class on Saturday evening. Saturday evening में हम लोग properties of cardiac muscle पढ़ेंगे नेक्स्ट क्लास में. आज हम इतने पे ही छोड़ रहे हैं, बारे में थोड़ा ध्यान से पढ़िए. आई एम नॉट गोइंग टू ओवरलोड यू विद मोर इंफॉर्मेशन. ओके.